Hi, welcome to the Molmatix. Today we are going to learn a very important cell organelle known as the ribosome. In this lecture, first of all, we will see the definition of ribosome. Ribosomes are the protein factories of the cell that produce proteins or polypeptide chains from the amino acids with the help of tRNA molecules. This is the ribosome molecule and this is the mRNA molecule. This ribosome translates code from mRNA molecule into the polypeptide chains with the help of tRNA molecules. This is the polypeptide chain. That means ribosome converts code from mRNA molecule into the polypeptide chain, and this is how synthesis of proteins takes place, or the protein synthesis takes place with the help of tRNA molecules. This is why ribosomes are known as protein factories of the cell. Ribosome is a non-membranous cell organelle. There are two types of cell organelles. One are membranous cell organelles and second are non-membranous cell organelles. Membranous cell organelles include mitochondria, endoplasmic reticulum. These cell organelles contains the covering of phospholipid bilayers. This is why these cell organelles are known as the membranous cell organelles. Non-membranous cell organelles like ribosome, cytoskeleton, they do not have covering of phospholipid bilayer. They do not have phospholipid bilayer. This is why these cell organelles are known as non-membranous cell organelles, and ribosome is a non-membranous cell organelle. Ribosomes are composed of rRNA and ribosomal proteins. In this structure, this brown colored molecule is the rRNA molecule, and this blue colored molecule is the protein molecule, or the ribosomal protein molecule. These ribosomal protein molecules and ribosomal RNA molecules interact with each other and form a complex molecule known as the subunits of the ribosome. There are two types of ribosomes, pre and the membrane bound ribosomes, and this is the endoplasmic reticulum. This portion is the smooth endoplasmic reticulum, because this portion of the endoplasmic reticulum do not have any type of ribosomes attached over it, and it looks a smooth surface. This is why this region is known as the smooth endoplasmic reticulum. But this is the rough ER, or the rough endoplasmic reticulum. It contains ribosome molecules attached over its surface, and it looks like a rough surface. That's why this region is known as the rough ER, or the rough endoplasmic reticulum. The ribosomes present over rough ER are known as the membrane-bound ribosomes. These are the membrane-bound ribosomes, whereas free ribosomes are freely present in the cytoplasm of the cell. This is the cell, and ribosomes present in the cytoplasm are known as free ribosomes. Then ribosomes are also known as ribozymes. This means enzymes which are made up of the ribosome molecules. Ribosome acts like a enzyme molecule. This is why they are known as the ribozymes, because the ribosomes perform catalytic peptidyl transferase activity that joins amino acids together to form a protein molecules. Then ribosomes are also known as ribonucleoprotein complexes, because at the molecular level, ribosome is a combination of RNA molecule and the protein molecules. Then the discovery of ribosome. The ribosomes were first discovered by George Emil Pallade in the 1955. He is the George Emil Pallade. He found ribosomes under electron microscope, and for his discovery he was awarded with the Nobel Prize in 1974. Then the discovery of structure of ribosome. The atomic structure of ribosomes were first given by Venkatraman Ramakrishnan, and he was also awarded by a Nobel Prize in 2009. He is the Venki Ramakrishnan. Overview of ribosome. Structurally, ribosome is divided into two subunits, small and large subunits. This is the small subunit and this is the large subunit. This portion is the large subunit. Small subunit holds mRNA molecule and large subunit holds tRNA molecules and produce polypeptide chains. These two subunits bind with each other and function as a single unit to create a proteins according to genetic code present in the mRNA molecules. Molecules associated with the ribosome like ribosomal RNA and ribosomal proteins are known as the translational apparatus because ribosome translates mRNA code from the mRNA molecule into the protein molecules. Structure of ribosome. Prokaryotic cells contain 70S type of ribosomes which are made up of 50S large subunits and the 30S small subunits, whereas eukaryotic cells contain 80S type of ribosomes which are made up of the 60S large subunits and the 40S small subunits. In prokaryotes, ribosome 70S type of ribosome and large subunit is 50S type of large subunit and small subunit is 30S type of small subunit, whereas in eukaryotic cells ribosome is the 80S type of ribosome, whereas 60S is the large subunit and the 40S is the small subunit. In the above examples, 70S and the 80S ribosomes contain S as a suffix. S is the Swedberg unit that indicates the particle size based on its rate of sedimentation under the acceleration. Process of protein synthesis. The process of protein synthesis takes place in the 4 stages, that is the initiation of protein synthesis. This starts with the reading of start codons. The elongation. This is the process where ribosome reads codons of mRNA molecule with the help of tRNA molecules. Ribosome and tRNA molecules together form a polypeptide chain of amino acids, and this process is known as the elongation. The process of termination is carried out by the ribosome by reading stop codons of mRNA molecules, and after the termination, recycling of this ribosomal apparatus takes place, and these are the 4 stages of the protein synthesis. The process of protein synthesis. The DNA segment of cell which encodes protein sequence transcribes its message on the messenger RNA chain. This is the DNA molecule and this is the mRNA molecule. DNA transcribes its code over mRNA molecule. Then this mRNA molecule is transferred into the cytoplasm towards ribosomes. This mRNA molecule binds with a small subunit of ribosome this mRNA molecules binds with the small subunit. This is the small subunit and this is the large subunit. And this is the mRNA molecule. This mRNA molecule binds with the small subunit. In case of prokaryotes, small subunit is the 30S, and in case of eukaryotes, small subunit is the 40S. The large subunit of ribosome binds with the tRNA and performs a catalytic function in the process of translation. Large subunit binds with the tRNA molecule, and tRNA molecule contains amino acid. These amino acids are joined together by the peptide linkage. That is the C double bond O NH. This is the peptide linkage. Large subunit binds with the tRNA molecule and performs a catalytic function. The small subunit reads the message of mRNA molecule and translates it into the polypeptide chain with the help of large subunit and the tRNA molecules. This is the large subunit and this is the small subunit. This small subunit binds with the mRNA molecule. This is the mRNA molecule. Small subunit binds with the mRNA molecule and performs a decoding function. The process of translation starts with the start codon, that is the AUG, and ends with the stop codons, that is the UAA, UAG and UGA. And these start and stop codons are only can be read by the ribosome molecule, and tRNA do not have anticodons for the start and the stop codons. This is why these two type of codons are only read by ribosome molecules. And this plays an important role in the initiation and process of termination. There are no tRNA molecules which can identify start and the stop codons. Only ribosomes can identify the start and stop codons. And these start and stop codons play an important role in the process of initiation and termination. These are the references. Thank you.